Hello and welcome to today's Journal Club on two papers that explore therapies for Rett syndrome. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research news and analysis. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this Journal Club if that material has already been published elsewhere or if you have first obtained express written permission from the presenter. Today's Journal Club will feature two presentations of recent research exploring therapies for Rett syndrome. Our first presenter is Rebekah Tillotson, who joins us today from Edinburgh, the United Kingdom. She is a member of Adrian Bird's lab at the University of Edinburgh and is pursuing a PhD investigating the role of MeCP2 protein function and Rett syndrome. She received her BSc in molecular genetics from the University of Edinburgh in 2012. She will present on her PhD research, which was recently published in Nature. Our second presenter, Laura Lombardi, joins us to discuss her postdoctoral work in Dr. Huda Zogby's lab at Baylor College of Medicine and the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital, where she works on discovering therapeutic entry points of the treatment of MeCP2 duplication syndrome. The work was recently published in Science Translational Medicine. Dr. Lombardi received her PhD in molecular and cell biology from the University of California, Berkeley, working in the lab of Dr. Jasper Ryan. She is currently a researcher at Tenaya Therapeutics in San Francisco, California. Welcome to you both.
Thank you.
Thank you. Okay, so hello everyone. Thank you for giving me the chance to present my PhD work to you all. So I've been working on a protein called MeCP2, which was originally discovered in Adrian's lab back in 1992, and it's been a main focus of his research ever since. We're interested in this protein for two reasons. The first is because it's an epigenetic reader of the DNA methylome, binding to both methyl CpG trinucleotides and methyl CAC trinucleotides via its MBD, which stands for methyl CpG binding domain. We want to know how MeCP2 interacts with methylome in neurons where it's most highly expressed. The second reason why we're interested in this protein is because it's been implicated in a range of disorders which are called MeCP2 related disorders. Loss of function mutations in hemizygous males result in neonatal encephalopathy. In heterozygous females, they result in Rett syndrome, which is characterized by 6 to 18 months of normal development followed by a crisis period where symptoms begin. These symptoms include loss of speech, loss of purposeful hand movements, autism, seizures, and breathing problems. And lastly, duplication of the MeCP2 gene also causes neurological disorders, particularly in boys, which Laura will tell you more about afterwards. So what does MeCP2 protein actually do? A survey of the literature will tell you that it does pretty much everything from transcriptional repression, transcriptional activation, chromatin compaction, alternative splicing, and microRNA processing. And to do all of these functions, it interacts with over 40 interaction partners whose interaction domains have been mapped throughout the protein length. So this gives you the picture that different bits of MeCP2 protein are all doing different things via different interaction partners. So I set out to ask which of these roles is the most important. Normally, to see which bits of a protein are most important, you would look at the evolutionary conservation. As you can see, MeCP2 is very highly conserved throughout its length throughout vertebrate species. However, if you look at where the missense mutations that cause Rett syndrome are, you get a very different picture, with a big cluster around the MBD and a smaller cluster in a domain called the ENCOL-SMART interaction domain, which picks out only these two domains as most important. This is backed up by the large number of neutral variants in the other regions of the protein, suggesting that the other regions of the proteins can be changed without losing function. So this brought us to the hypothesis that MeCP2 functions as a bridge between methylated sites on chromatin and the ENCOL-SMART complex, and you only get Rett syndrome if you break this bridge, with the MBD mutants breaking it the bridge between MeCP2 and chromatin, and the NID mutants breaking the bridge between MeCP2 and the NCOS mark co-repressor complex. So I set out to test this hypothesis, and I did this by making a series of truncated versions of MeCP2, and then to go on and test whether these proteins function in MeCP2 knockout mice. So I have 3 truncations. The first is missing just the N-terminus of MeCP2, the second is missing both the N and the C, and the third is missing the N-C and the intervening domain between the MBD and the NID, which I've replaced with an exogenous NLS from SB40. I've also tagged all of these proteins with GFP. So first I'm going to tell you about the 2 larger truncations, delta N and delta NC. Analysis of their protein levels in homozygous male mice shows that they're expressed at approximately normal levels, so this won't affect the phenotype of the mice. So what do the mice actually look like? The delta N mice was scored for over a period of a year, and in contrast, which I've shown in grey, to MeCP2 null mice, they don't show any phenotypes and are indistinguishable from their wild-type littermates throughout the whole year. They have no weight differences, and all 10 of them survive throughout the year. The delta NC mice also show no phenotypes throughout the year. They are slightly heavier than their wild-type littermates, and again, all 10 of them survive throughout the year. I had a second cohort of back-cross mice which underwent a series of behavioral tests. So the first is looking at hypoactivity with the elevated plasmase, and there was no difference in the distance traveled between the delta N mice and the wild-type littermates. They had no anxiety phenotype in either the open field test measured by the amount of time spent in the central region, or the elevated plasmase measured in the amount of time spent in the open arms of the test. And then lastly, they showed no motor defects on a 3-day protocol of an accelerating rotor rod. So the delta NC mice underwent the same series of tests. They had no hypoactivity phenotype in the open field test. They had a small but significant phenotype suggesting decreased anxiety in the open field test, shown by spending more time in the central region, but this wasn't recapitulated in the elevated plasmase, and again they showed no phenotype in the accelerating rotor rods test. So overall, the N and C termini of MeCP2 are broadly dispensable for protein function. So what about the middle? So this is the smallest of the truncated proteins, where the N-terminus and C-terminus and intervening domain have all been deleted. Analysis of the levels of protein in the brains of homozygous male mice here showed that the protein levels were down by about 50%. This wasn't true at the RNA level, so it looks like this protein is unstable. So this was unexpected, and we need to take this into account when interpreting the phenotype. The first cohort that underwent was observed over the course of a year, showed mild symptoms, mostly gait abnormalities and hind limb clasping, with an average score of about 2 to 2.5. They're about 40% lighter than their wild-type littermates, and 9 out of 10 of them survived over the course of a year, with 1 dying of unexplained circumstances at 43 weeks. Second cohort that underwent behavioral tests showed no hypoactivity in the open field test. They showed decreased anxiety in the elevated plasmase, shown by decreased time spent in the closed arms, but this wasn't recapitulated in the open field test. And most strongly, they showed impaired motor function on the accelerating rotor rod, with decreased performance over the 3 days, making them significantly different from their wild-type littermates by day 3. So they aren't completely normal, but they're obviously much better than the nulls that I've got here for comparison. So we went on to look at the therapeutic potential of this truncated protein. Back in 2007, it was shown in Adrian's lab that delayed activation of wild-type MeCP2 gene can reverse neurological symptoms in MeCP2 deficient mice. So I wanted to ask whether the delta NIC allele could also cause symptom reversal. So this was done the same way as back in 2007, where you have a flocked stop cassette, so it's a stop cassette flanked by 2 LOXP sites with an intron 2 of the MeCP2 gene, stopping transcription of the MeCP2 gene. These mice are bred with the Cre-ER transgenic line, so this pre-recombinase is kept in the cytoplasm until the mice are injected with tamoxifen. The Cre goes into the nucleus, excises the stop cassette, reactivating the gene. So I used this allele, but instead of having the wild type, I had my delta NIC allele. And this is the result. So as you can see in purple and in red, the mice with the stopped allele develop symptoms from weaning at 4 weeks, going up to about 6 weeks when the tamoxifen injection regime began. The ones carrying the Cre-ER, the symptoms plateaued and then reduced to about 2.5, so similar to the constitutively expressing delta NIC mice, and all of them survived for 28 weeks. In contrast, the stop controls that do not carry the Cre-recombinase continue to develop symptoms and are dead by 6 months of age. So what about a therapy that we can actually administer to patients? So for gene therapy, we're thinking about the advantages of having a small allele given the limited capacity of gene therapy vectors. The current gold standard for gene therapy is produced by Stuart Cobb's lab. They use short complementary AAV2 genomes packaged into AAV9 caspids. They have made a promoter which takes out 426 base pairs of the mouse MeCP2 promoter, as well as selected features from the 3' UTR. They use direct brain injections at P1 or 2 at a dose of 10 to 11 viral genomes per mouse. So this is a schematic of their vector. If we were to change it for the delta NIC allele, you can see that it's much smaller and there's scope to add better regulatory sequences. But first we wanted to test the humanized delta NIC with the same conditions optimized for the wild-type vector to see whether it worked just as well. And as you can see, it does. So in purple, we've got the null mice injected with the humanized delta NIC, which have reduced symptom severity compared with the nulls shown in gray and much prolonged survival. We also have limited adverse effects from the wild-type mice injected with the humanized delta NIC. So there's very little toxicity caused by overexpression, which is a big problem in these studies. So to conclude, my findings strongly support our hypothesis that MeCP2 functions as a bridge between methylated chromatin and the encosmart co-repressor complex. And this is exciting because the regions that I've deleted contain several binding domains. For example, the heterochromatin protein HP1 binds to the N-terminus of MeCP2, and the microRNA processing protein DGCR8 and 3 splicing factors have all been reported to bind to the C-terminus of MeCP2. However, the delta NIC mice do have a bit of a phenotype, which might be due to their reduced levels. So the next thing to do is to overexpress delta NIC to reach wild-type levels and ask whether that's the cause of their phenotype. Nevertheless, the delta NIC allele has therapeutic potential, and so we want to go on to optimize this vector utilizing the extra space that we've made. So I'd like to thank you all for listening, thank everyone in Adrian's lab, and also, of course, Stuart Cobb's lab, who did the gene therapy work. And I'll now hand you over to Laura.
Hello, I'm Laura Lombardi. Thank you very much to Claire and Spectrum for having me. I recently finished my postdoctoral work in Dr. Huda Zogby's lab at Baylor College of Medicine, and I'm excited to be here today to talk to you about our paper. As you heard from Rebekah, Rett syndrome results from loss of function mutations in MeCP2 as discovered by the Zogby lab in 1999. However, an increase in MeCP2 also results in a devastating neurological disorder, MeCP2 duplication syndrome. It was discovered in 2005 and is consistent with earlier work in mice from our lab and others. MeCP2 duplication syndrome is characterized by hypotonia, intellectual disability, as well as autistic features and epilepsy. It explains around 1% of X-linked intellectual disability, and approximately 40% of males die before the age of 25. As would be expected with a dose-sensitive gene, triplication of the locus results in more severe symptoms than duplication, and both of these are well modeled by both TG1 and TG3 mice. Dr. Zogby and her lab are committed to understanding and developing treatments for this disorder using the mouse lines that first suggested it may occur in humans. So mouse models exhibit acute sensitivity to the dose of MeCP2, mirroring the human situation. Male mice, as you can see here, that express twice the amount of MeCP2, indicated here as MeCP2 TG1, are approximately as sick as mice that express less than the normal levels or half the levels of MeCP2, as indicated in the blue dots. Additionally, you have further phenotypic severity if the expression of MeCP2 is overexpressed threefold, as in the TG3, or there's no MeCP2 at all, indicated here in the black dot. This really suggested to us that one of the best ways to develop potential therapeutics for MeCP2 duplication syndrome was to find genes that modulate MeCP2 levels. Now, of course, for therapy to be effective in treating the disease as opposed to the symptoms, these disorders have to be reversible. And as Rebekah mentioned, this is shown to be the case with respect to both Rett and MDS models, as shown by both the Bird and the Zogby labs. They can be rescued after symptoms emerge by restoring MeCP2 levels. Despite this, there is currently no FDA-approved therapies available to clinically modulate MeCP2 abundance. So for a protein whose levels must be tightly regulated, little is known about factors that affect MeCP2 turnover or stability. Thus, we hypothesized that there might be multiple endogenous regulators of MeCP2 stability and designed a screen to assess this. So the goal of this study was to really establish a robust screening platform for regulators of MeCP2 stability by generating a MeCP2 protein level reporter cell line, as well as optimizing secondary and tertiary screens, and validating in vivo with a mouse model of disease. And this pipeline was established so that we could then extend the realm of candidates we were looking at to the whole druggable genome. So to undertake this arrayed screen for regulators of MeCP2 stability, we started by developing a reporter cell line. This is a bicistronic reporter transgene which allows for monitoring of MeCP2 stability by a C-terminal fusion of GFP while controlling for variation in transgene expression by normalization to the independently translated ds-Red. With this reporter in hand, we performed an arrayed siRNA screen against all known amputative human kinases and phosphatases by flow cytometry analysis, being able to determine the ratio of MeCP2 fused to GFP over ds-Red, such that things that acted to stabilize MeCP2 when inhibited would result in a decreased GFP to ds-Red ratio. To control for variance in transgene expression, as mentioned, we assayed the ratio of GFP to DS-Red for each siRNA and compared it to the effect of 3 scrambled on-plate control siRNAs. We identified 480 siRNAs that significantly affected the GFP to DS-Red ratio, as indicated here on the top. To enrich for true candidates to follow up, we required that 2 out of the 3 siRNAs targeting a candidate gene significantly affected the MeCP2 GFP over DS-Red ratio in the same direction, and that the candidate gene be expressed in the brain. And then we performed a secondary screen using independent siRNAs of those candidates to limit off-targeted effects. And not only did we repeat this with the original MeCP2 GFP cell line, we also repeated it in parallel with a DS-Red iris GFP control cell line to basically determine which of these effects were specific to MeCP2 as opposed to simply affecting the iris GFP. Next, we performed a tertiary screen in which we made 3 significant changes. One is that we switched cell lines to confirm that this took place in an independent cell line. Additionally, we started monitoring endogenous MeCP2 as opposed to the overexpressed fusion protein. And thirdly, importantly, we determined if the candidate was actually being knocked down. So previously that had not been addressed. So what you can see if we start here on the bottom left, where we're looking at the RNA expression of the candidate PPP2R1A, that only shRNAs A and C actually result in significant knockdown of the gene. Additionally, if you look above, where we're looking at the level of endogenous MeCP2, you can see that only when we have a significant knockdown of PPP2R1A do we see a significant decrease in MeCP2 levels. And so depicted as well are candidates which displayed a similar correspondence, such as HIPK2 and also HIPK1 and REOC1. We were actually very interested to obtain HIPK2 and HIPK1, as HIPK2 is a known kinase of MeCP2. Since HIPK2 was a known kinase of MeCP2, we wanted to determine if HIPK1 also was, as they are known to share some substrates, and whether they phosphorylated the same published site, which is phosphoserine 80. So on top, what you're looking at is a positive control in vitro hot kinase assay, in which in lane 1 we have recombinant HIPK2 as well as MeCP2 fused to CPP, incubated with radioactive ATP. And what you can see is that indeed MeCP2 is radioactively labeled as well as HIPK2 autophosphorylation. Additionally, in the absence of kinase, we see no radioactivity of MeCP2. To control for potential phosphorylation of the epitope, we also bound the reaction exhibited in lane 1 and then washed off the kinase. So you can see a decrease in the Western blot of both HIPK2 and also its activation loop phosphorylation. And then we cleaved off the tag, and you can see MeCP2 migrating here with the radioactivity, as well as you can see the MeCP2 size migration in the Kamasi. And so on the right, you can see that additionally, this was true as well for HIPK1. So we are very interested to know if they were phosphorylating the same residue. We were surprised to find in data I'm not showing here, but we were surprised to find that if we provided MeCP2 that could not be phosphorylated on serine 80, we still obtained radioactivity when HIPK1 or HIPK2 were provided. Thus, we performed mass spec on these in vitro kinase reactions for an unbiased approach, and we discovered that the main phosphorylation that was occurring in these in vitro reactions was actually phosphoserine 216. And you can see the mass spectrum here, as well as the percent peptides that were phosphorylated. So this was exciting to us because phosphoserine 216 had been previously identified in vitro but was an orphan phosphorylation. So the first thing we did was, as you can see on the left, was to assess if this was actually a conserved site. And as you can see, comparing to serine 80, serine 216 is also very highly conserved in vertebrates, and the HIP consensus site is indicated here as well. So the next thing we did was to see if we could actually demonstrate this site in vivo in the mouse brain. And so for this experiment, we performed IP mass spec, and we were able to detect this phosphorylation in the mouse brain. So moving forward, knowing that it occurred in vivo, we wanted to generate a phospho-specific antibody, the control of which is here, and this allowed us to detect this phosphorylation in cells in a HIPK2 and HIPK1 dependent manner, with a similar increase upon overexpression of HIPK2 and HIPK1 as in the phosphoserine 80. Given this, we wanted to probe the functionality of the phosphorylation. So we generated stable cell lines so that we could turn off the expression of either wild-type MeCP2 or MeCP2 that was mutated to the phosphomimetic form, serine 216, to aspartate. And turning off the expression, we could then compare the relative half-life of wild-type versus the phosphomimetic form. And as you can see here, the constitutive phosphorylated state stabilizes the protein. Consistent with that, if we look at HIPK2 mutant brains, we see a decrease in MeCP2 levels, suggesting that both HIPK2 and potentially this phosphorylation are acting to stabilize MeCP2 levels. So as our proof of principle candidate to move into disease models with, we selected protein phosphatase 2 regulatory subunit 1 alpha, which is depicted here. So this is a scaffold for protein phosphatase 2A, which functions as an obligate dimer, which is indicated here between the catalytic subunit and the scaffold subunit. The most abundant isoform of the scaffold is encoded by PPP2R1A, resulting in 90% of the scaffold present in cells. So additionally, we saw on the primary screen that we got multiple hits for this complex. So to determine the effect of PPP2R1A knockdown in the mouse brain, not dissimilar to what Rebekah discussed, we optimized a viral delivery system in which AAV8 containing shRNA was injected into the lateral ventricles of neonates, resulting in widespread cortical and hippocampal infection. So to perform this experiment, 2 different AAV vectors were utilized, one encoding shRNA against the candidate, while the other encoded scrambled shRNA. And what you can see from this in vivo experiment, you can see that that the decrease in MeCP2 occurs, and that this decrease is significant and corresponds with the extent of PPP2R1A knockdown. Additionally, in a separate cohort, we tested directly on the same lysates. We isolated protein and RNA and showed that this decrease in MeCP2 is happening at the protein level specifically without any decrease in MeCP2 RNA. So consistent with the design of the screen, this regulation of MeCP2 is happening post-transcriptionally. Additionally, since the holoenzyme of PPP2A works as an obligate dimer between the scaffold and the catalytic subunit, we hypothesized that this decrease was due to a decrease in the enzymatic activity of protein phosphatase 2A. Thus, we tested this directly by using known chemical inhibitors of protein phosphatase 2A, okadaic acid, and phosphorylation. And this is in cultured primary neurons. And what you can see here is we indeed recapitulate this regulation by protein phosphatase 2A. So because of the ability to inhibit this enzyme, we then moved in vivo, both in the wild-type situation and also in the mouse model of MeCP2 duplication syndrome, TG1. We moved in vivo, and we were able to show that indeed okadaic acid similar to the genetic disruption of protein phosphatase 2A, results in decreased levels of MECP2 as well as an independent inhibitor, phosphorylated. Thus, at this point, we felt that we had firmly validated protein phosphatase 2A as a regulator of MECP2, given we had genetic evidence as well as 2 different inhibitors resulting in this effect. So with phosphorylated, we saw a similar decrease in MECP2 levels, and so we moved forward to assess behavioral analysis with phosphorylated. Even though it's less potent than ocadic acid, it is more specific of an inhibitor. So what we did is, as Rebekah introduced you to, we used the accelerating rotating rod, or rotor rod, and we tested the ability of the decreased MECP2 levels in the duplication model mice to rescue the abnormal motor activity of the TG1 mice. And so you can see that phosphorylated treatment in the TG1 mice partially rescues the persistence on the accelerating rotating rod, as evidenced by a decreased latency to fall compared to TG1 mice treated with saline here in red. So conclusions of what I've shown you today are that this screen allowed for the identification of 4 novel modulators of MECP2 protein levels, and that all 4 of the modulators regulate MECP2 at the post-transcriptional level, with HIPK1 and HIPK2 directly phosphorylating MECP2, as well as phosphoserine 216 stabilizing MECP2, consistent with the decreased MECP2 we see in the HIPK2 mutant mice. Additionally, I showed you that both genetic and pharmacological inhibition of protein phosphatase 2A decreases MECP2 levels in vivo, and that this was able to rescue some of the motor abnormalities in the mouse model of MECP2 duplication syndrome. So ongoing work on this project is a lot, further characterizing the functional significance of the phosphoserine 216, as well as assessing in the HIPK1, HIPK2 double mutant mice, or double heterozygous mice, to assess redundancy and genetic interaction with the TG1 mice. Additionally, the REOC mutant mice have been generated and are being assessed currently for regulation of MECP2. And as I mentioned, this entire study was designed to really test the pipeline and the feasibility of using this reporter. And now that we have confirmation in vivo, we're moving forward with a genome-wide screen for MECP2 regulators using this stability reporter as well as this pipeline. So I'd like to thank you all very much for listening, as well as Huda Zogby, who's an absolutely amazing mentor, as well as my collaborators in lab, as well as extramural collaborators. So thank you so much. We look forward to your questions.
Okay, thank you so much, Becky and Laura. We do have a couple of questions from the audience. The first one is directed towards you, Becky. Since mice with a lack of the amino terminus of MECP2 do not exhibit a phenotype, does this suggest that there are no functional differences between MECP2E1 and MECP2E2?
Okay, so I should give a bit of background. There are 2 isoforms of MECP2 called E1 and E2. The only difference between them is the very extreme N-terminus. So most of the protein is encoded in exons 3 and 4, and then E2 has a short peptide from exon 1, and E2 has a short peptide from exon 2. Because of the structure of the gene, I don't know if I'm able to go back to my slides.
You can.
Does that work?
You can.
Okay.
Yeah.
So because of the structure of the gene, we're only really able to target exons 3 and 4. So as you can see, my truncated proteins do actually have the tails from either exons 1 or 2, and then that gets fused straight onto the MBD. So I don't think that I've really addressed whether the either of the peptides, either the E1 or the E2 peptide, are dispensable for function because I wasn't able to get rid of them. What I can tell you is that the only different role that I am aware of between the two isoforms is that E2 has been reported to bind to FOXG1 and E1 hasn't, with the interaction domain covering this beginning region. So what I don't know is whether taking out the chunk between amino acids 12 and 72 disrupts that interaction. Whether I've got rid of that interaction, I haven't established yet.
Okay, and for Laura, is S216 close to the IND domain of MECP2?
Okay. So yeah, is that for the in-core interaction domain?
Mm-hmm.
Okay, yeah, so the in-core interaction domain, Becky, you can help me out. I don't know what amino acid it's precisely starting at, but you can see 216 is in the TRD. But yes, not precisely regulating the NID, no.
So
I should say we do speculate that it might be due, yes, to potential different protein interactors, and that could be affecting stability, since MECP2 appears to be quickly degraded if it's not stably associating with chromatin.
Yeah, so the 216 is in the intervening domain. So the NID is the C-terminus of the transcriptional repressor domain. It's just the last few amino acids of that domain. So yeah, it's not in the NID, but it is in the transcriptional repressor domain that was previously mapped.
Okay. Directed towards you, Becky, did you look at sex differences between male and female mice, or were you using all male mice?
I only used male mice because we predicted, and we were correct, that the phenotypes would be very mild, and so within a year we wouldn't expect to see any differences in the female mice. Because even with us 133C mice, the phenotypes are still quite mild by 1 year of age, so it was to give the most sensitive function, assay of protein function to use the hemizygous males.
Does the mini-gene approach, so the approach that you used, is there a critical period for it, or can it be used in adult and juvenile mice and in vitro?
So the latest I've gone is 6 weeks. So when I started the tamoxifen injection, so it's 6 weeks, and I saw symptom reversal. So that was, I'll just go back. Yeah. So the mice were injected, so sort of young adults at 6 weeks, and then the symptoms were reversed. With males, I couldn't have gone much later because they were getting quite sick by that point, but I would predict with females that you could treat them as adults. It's just, obviously, with the premature death phenotype in the males, you need to catch them before that.
Mm-hmm. Do you have any next steps for this particular approach that you're looking to pursue?
So therapeutically, the next step is to further develop the vector, use the space that's created by getting rid of the dispensable domains to lead to better regulation of the protein, so that you don't, because a big problem with gene therapy studies is overexpression.
Mm-hmm. So you have that narrow window of opportunity.
Yeah. Yeah, so we want something that can be better modulated. Yeah. So as Laura's introductory slide showed, that the levels are really, really important, and gene therapy using viral vectors doesn't have very much control for the levels of protein that you get.
Laura, for your for the drug identification screen, when you're looking at which genes to screen, is that targeted or are you going in agnostically?
Yeah, so for this study that I talked about, we went after kinases and phosphatases first, given that they're often druggable, and also obviously MeCP2 has many phosphorylation sites, and they're they're very likely to regulate post-translational stability. For the broader, quote-unquote, genome-wide druggable genome screen, sort of biotech and pharma have an established class of what they consider druggable, so certain classes of genes, particularly enzymes and receptors, et cetera, for which there are sort of tool compounds available. So this current library was curated in collaboration with Stephen Elledge at Harvard, as well as various consultants in industry to help us basically narrow in on things to not just determine regulators, but once we've found that regulator, be able to move forward with it.
So the next step of looking at the sort of screening 7,000 for the genes, that's a more agnostic approach.
Yeah, so this druggable genome, current broadened druggable genome screen is 19,000 genes, so many, the majority of the genome, yes.
When we were reporting on this story, Dr. Zogby talked about the idea that there should be a combination approach to treatment of MeCP2 levels. And this is a question for you both, is there the possibility of combining both drug treatments with other approaches such as gene therapy treatments in the future?
I think the biggest... we just both answered.
Go ahead, Rebekah.
I think what would be a nice thing to do is if you do put too much MeCP2 with gene therapy, to be able to use drugs to modulate it back down again. And obviously it's nice that in your screen you have found targets that reduce protein levels.
Yeah, so I should, multiple points. One is that the idea for doing combination therapy really has to do with what targets you have, which regulators you have. If those candidates have multiple substrates, for example, you want to be very careful with how much you can inhibit them if you don't want to get pleiotropic results. And so often the best thing to do is to do a two-hit situation where you're hitting two different regulators at low levels such that you're not compromising their own, sort of endogenous or other substrates too severely. That way you try to keep it as targeted as possible such that they're converging on MeCP2 regulation, I believe is Huda's vision for combinatorial therapy. I should also note that even though in this discussion I was just talking about things that, when inhibited, decrease MeCP2 levels, we also find things that stabilize MeCP2 levels. And this is this is something that could be used in, collaboration with the work Rebekah was discussing, in which, we could supply something which would then help to stabilize the delta NIC construct. And these are regulators that we think are potentially also very useful for certain hypomorphic Rett alleles, which really just need a boost in expression. For example, the T158M.
Okay, thank you. That's all the questions that we had for today. So I will say thank you to you both and thank you to everyone for participating. A complete replay of these presentations will be available on the Spectrum news site in a couple of days. The Journal Club is part of an ongoing series, so please check our site for upcoming talks to be announced soon and for video archives of previous webinars. Our next webinar is in January with James McPartland on autism biomarkers. And thank you again, Laura and Becky, and thank you all for tuning in.